So now we've discussed sort of what happens at the fine level of detail. Let's for a second take a step back and look at what happens regarding the replication fork. I had said that there was a single replication fork for prokaryotes, whereas for eukaryotes there's multiple origins of replication. In this example here, we're going to consider this as a prokaryotic replication fork. So shown in part 1 of this figure is double-stranded DNA. It's anti-parallel because the 5' end of one strand is associated with the 3' end of the complementary strand, and the 3' hydroxyl is associated with the 5' phosphate of the other at the other end. And of course it's complementary because these rungs of the ladder, if you will, are representing the hydrogen bonding that happens between the A's, the T's, and the G's, the C's. So there has to be an initial unwinding event that breaks some of these hydrogen bonds. The job of unwinding the DNA goes to DNA helicase. DNA helicase is an enzyme that initially comes in, it cleaves some of the hydrogen bonds, and it opens up a little bubble, which we call the origin of replication. There are specific DNA sequences where this happens. You don't have to worry about them, but as long as you understand that DNA helicase is going to unwind at the origins of replication, well, then you're fine. So what's happening at the replication fork? What's happening is that it's moving bidirectionally. Remember that little fact, it's bidirectional, so the replication fork is going to be opening from right to left. It's also going to be opening up from left to right in both directions. Let's just consider one portion of the replication fork. To make this explanation a little bit easier to accomplish, I want to just focus on one direction of the replication fork, with the understanding that the same exact thing is going on at the other end of the replication fork because it's bidirectional. So there's going to be 2 strands of the DNA. Depending upon the orientation of the 5' and the 3' group relative to the direction the replication fork is moving, there's either going to be a leading strand or a lagging strand. So let's first consider the leading strand. The leading strand is long and continuous, and think of it as going into the replication fork. It's going in the same direction. It's being synthesized or duplicated in the same direction as the replication fork is going. What do I mean by that? Well, here we have this little arrow, which is telling you that the replication fork is moving from right to left, and we have 2 strands of DNA. One is being duplicated from right to left, and one is being duplicated from left to right. The strand that's being replicated from right to left is moving in the same direction as the replication fork, which is also moving from right to left. Therefore, we refer to that as the leading strand. Now, why is that long and continuous? Why do we call it a leading strand? It has to do with the rules of DNA replication. Remember, the polymerase has to read a template from 3' to 5', and it has to synthesize a copy from 5' to 3'. As this replication fork opens up even more, this template can continue to be read from 3' to 5'. This product can continue to be synthesized from 5' to 3'. What does this little yellow block represent at the 5' end of the DNA? That's the RNA section. Remember, RNA primase had to lay down a little RNA primer in order to get DNA polymerase going. So, for leading strand synthesis, the primase synthesizes the primer from 5' to 3'. That would be this right here. And then in eukaryotes, there's a polymerase alpha and delta. Polymerase alpha and delta in eukaryotes, there's polymerase alpha and delta, which are going to extend this in the 5' to 3' direction, moving into the replication fork. And lastly, helicase will continue to unwind the double-stranded DNA here at the fork. Now, let's take a peek at lagging strand synthesis. I think you already knew that because there's a leading strand, there's also going to be a lagging strand. So for lagging strand synthesis, you have down here discontinuous synthesis. The DNA is moving away from the replication fork. It's going to be made in short segments. Why is that? Why does it have to be made in short segments? Why can't the DNA just be read as I'm drawing here and a new product laid down in the same direction? Why can't that happen? It's because of the rules of DNA replication. Remember, DNA always has to be read from 3 to 5 prime, and new product has to be laid down from 5 to 3 prime. So, in other words, we have to read this from left to right. But the problem is, this is still closed up. That's inaccessible, double-stranded DNA that has yet to be unwound. So when the replication fork opens up, a short RNA primer is going to be laid down. The template's going to be read in the 3 to 5 prime direction, and the polymerase is going to synthesize from 5 to 3 prime. Now, the polymerase can't get to the sequences that it needed to, right, down here, but it can get to the sequences here because the replication fork is opening up. So a primase will lay down a short RNA primer, it'll read from 3 to 5 prime, it'll synthesize from 5 to 3. This will open up, it'll read from 3 to 5, it'll synthesize from 5 to 3. It'll do the same, etc., etc., etc. It'll make these in short little fragments. And what do we call these fragments? We call them Okazaki fragments, okay, named after the scientist who sort of discovered them. So Okazaki fragments are the short little fragments that are seen on the lagging or discontinuous strand. Eventually, these will all be joined together and the DNA will look like it should, but as the DNA is being polymerized, there's a leading strand and a lagging strand. After the leading strand is, or I should say, while it's still being synthesized and while the lagging strands are still being synthesized, there's a special RNase, and it's called RNase H. RNase H. It has a special nuclease activity called 5' exoribonuclease activity. Sort of a low-yield fact for your exam. But what this exonuclease activity does is it comes in and it removes these RNA primers. It removes these RNA primers and it fills in the gaps with DNA. And actually another enzyme called DNA ligase will form the last phosphodiester bond to help make this into a continuous strand of DNA instead of the discontinuous Okazaki fragments that we saw earlier in the process. This would have to happen for every single RNA primer that gets laid down. To help you visualize this isn't in your text if you're following along in your textbook. Here's a close-up that illustrates what's going on at the replication fork, at one side of the fork. I'm going to tell you in advance that the fork is advancing from left to right. It's advancing from left to right. With that information in mind, you should be able to tell me which of these strands is leading and which of these strands is lagging. So if the replication fork is advancing from left to right, which strand is the leading strand? It has to be the strand that reads from 3 to 5 prime in that direction. Because if this strand reads from 3 to 5 and the replication fork is advancing in that direction, well, then you could simply start laying down new complementary anti-parallel DNA and following that replication fork. As these open up, the template's going to be read by the polymerase and these will be synthesized. That's a leading strand. The bottom strand, therefore, would be the lagging strand. Why? Well, it has to be read from 3 to 5 prime. But these bases here are inaccessible at this point. So the best it can do is to access right here at the replication fork and read from 3 to 5 and synthesize the product from 5 prime to 3 prime. That's the lagging strand. It's going to open up a little bit more. It'll start reading from 3 to 5 and synthesizing from 5 to 3. It'll read from 3 to 5, synthesize 5 to 3, over and over again until eventually the Okazaki fragments will be linked together through that RNase H activity. At least in eukaryotic cells, you see that RNase H. A couple of accessory enzymes. Don't forget about DNA helicase. DNA helicase is going to be what unwinds the DNA at the replication fork. So that's going to be what separates these hydrogen bonds. There's a topoisomerase as well, and topoisomerase is going to be downstream of the replication fork, sometimes upstream, and it's going to relax the tension in the DNA. You see, being double-stranded, if you start separating the DNA at certain points, it's going to create tension at downstream points at the DNA. That tension has to be relieved, otherwise the DNA is either going to snap back together or it's going to break, and it's going to become fragmented. The job of topoisomerase is to cleave bonds, unwind the DNA, and then reform those bonds. So topoisomerase activity, or swivelase, if you want to think of it that way, helps relax the tension on the DNA. Why am I telling you this? Because there's a clinical connection, right? There's an important clinical connection we'll be making in a minute. So, to reiterate the steps involved in DNA replication, the origin of replication in prokaryotic cells, there's going to be one single, what we call ORI site, that stands for origin, on each of those bacterial chromosomes. Most bacteria have one chromosome. In eukaryotic cells, there's multiple origins of replication, or ORI sites, on each chromosome. How is the DNA helix unwound? Well, it's the job of DNA helicase for both prokaryotic and eukaryotic cells. How is the template strand stabilized? Well, here's an important protein that we'll discuss. It's called single-stranded DNA binding protein, and it's effective in both prokaryotes and eukaryotes. Single-stranded DNA binding protein, or SSB, is a protein that will bind to, as the DNA fork opens up, the DNA becomes single-stranded. There's something interesting about single-stranded DNA in the cell. There's two things. Number one, it wants to snap back together. It's energetically more favorable for the DNA to be in the double-stranded DNA conformation. That's number one. Number two is single-stranded DNA is susceptible to degradation. There's enzymes that would just love to come in and start degrading that single-stranded DNA. So how does the cell get around this? Well, there's single-stranded DNA binding proteins that, as the DNA separates, these proteins bind to each of the single strands, and number one, they protect it from degradation, and number two, they stabilize the DNA in the single-stranded conformation. So keep that in mind. How is the RNA primer made? In both cases, prokaryotes and eukaryotes, you see that there's a primase activity that lays down some RNA prior to the DNA coming aboard and extending. The synthesis of DNA, there's going to be a leading strand and a lagging strand. The lagging strand, as we said, is divided up into Okazaki fragments. Here's the difference that you need to keep in mind. In prokaryotes, it's DNA polymerase III that does most of the replication. Not all, but most of the replication is accomplished by DNA polymerase Roman numeral number 3. In eukaryotes, the DNA replication is accomplished through DNA polymerases alpha and delta. And actually, there's a little bit of controversy over which types of DNA polymerases are involved here. I don't think it's extremely likely you'll be asked about specific isomers of eukaryotic DNA polymerase, but in case you are, what you would be asked about is DNA polymerases alpha and delta. Those are the ones that are involved in eukaryotic replication. Furthermore, in prokaryotic cells and in eukaryotic cells, the primers have to be removed. In prokaryotes, there's a DNA polymerase I activity that does this. It has the 5' to 3' exonuclease activity, which can gobble up. It can gobble up the nucleotides as it lays down new nucleotides. Whereas in eukaryotes, there's an RNase H enzyme, which does the same thing. Lower yield enzyme. Replacing of RNA with DNA. Once again, in prokaryotic cells, how do we take that RNA primer that was there and replace it with DNA? Well, thankfully, we have something called DNA polymerase I that can do the job for prokaryotic cells. For eukaryotic cells, it's unknown. And you gotta love that, because anytime something's unknown, guess what? It's one less thing for you to know, right? Okay, so it's unknown. 5 years from now, there may be something in this box. How are the Okazaki fragments joined? It's by DNA ligase for both prokaryotes and eukaryotes. I want you to focus some attention down here, because removal of positive supercoils ahead of the advancing replication fork is an important mechanism. Why is it important? Because if you block that mechanism, you can kill off the bacteria. How do I mean? Well, your fluoroquinolones that you're very familiar with. These would be ciprofloxacin, moxifloxacin, levofloxacin, gadafloxacin. Any type of quinolone drug is going to specifically block prokaryotic replication, and the way it's going to do this is through inhibition of DNA gyrase. Now, DNA gyrase is just a fancy name for DNA topoisomerase 2. When you're in prokaryotes, you call it DNA gyrase, but it really functions like DNA topoisomerase 2. In eukaryotes, we also have topoisomerases to remove these positive supercoils, and actually, there's a couple of drugs that will target eukaryotic topoisomerase. You would use these obviously to treat some types of cancers, and the drugs' names would be like etoposide or tenofovir. These are drugs that specifically target eukaryotic topoisomerase II. You don't need to worry about the differences between topoisomerase I and topoisomerase II. Not too big of a deal. And actually, in some autoimmune diseases, you're going to see antibodies produced against topoisomerases. Scleroderma comes to mind. So you're going to see these topoisomerases from the point of view of understanding their mechanism of action. They remove positive supercoils. You're going to need to know antibiotics that target their activity, so the fluoroquinolones, and then you're going to need to know about autoimmune diseases like scleroderma, where you get autoantibodies produced against these topoisomerases. Finally, synthesis of telomeres. Telomeres synthesis is not required in the prokaryotic cell because why? They're not linear chromosomes, they're circular chromosomes. Telomeres are required in certain germ cells and in cancer cells, in eukaryotic cells, and they're required because the DNA is linear in structure.